Okay, everyone. Thank you very much for joining our first Ask a Researcher of 2024 here. It's fantastic to see such a large group of you. Thank you so much for taking the time to come out today. So I'm just going to give you a quick rundown of housekeeping, and then I will hand over to our presenter. So we've got Professor Ann Morgan here with us today, who's going to talk about improving outcomes for vasculitis patients, and we'll have various stop points along the presentation for you to be able to ask any questions. Okay, so yeah, thanks again. Lovely to see so many of you in person, meeting properly at last. And I will pass over to Professor Morgan. Thank you.
Thank you, Amy. Can everybody hear me at the back? Okay, because I don't think there's a microphone here. So hello everyone, I'm Ann Morgan. I'm one of the rheumatologists here in Leeds. And I'm going to talk through some aspects of some of the research that we're doing here in Leeds through the Biomedical Research Centre. So I thought I'd just start by saying something a little bit more broadly about our musculoskeletal theme. So for many of you who've been attending the Ask the Researcher sessions in Leeds for some time, you'll know that our Biomedical Research Centre was entirely focused on musculoskeletal disease. Well, with the last round of applications, we were very successful, and we've managed now to expand into a whole number of areas. So rather than just have the one musculoskeletal theme, we now have 6 different themes. So this is a huge, this is fantastic for Leeds, and it means we can really extend a lot more of this early translational research right the way across medicine in Leeds. So there's a whole series of those. And in terms of musculoskeletal disease, it's still very much the bulk of the BRC. So for all the other areas, the maximum number of disease areas that they're covering is 3 different disease areas, whereas within musculoskeletal, we have 7 what we call work streams, so 7 different areas that broadly cover different rheumatological diseases. And there's also an orthopedic section within surgery. So musculoskeletal in general covers, over half of the BRC. And I'm the lead of, sorry, this isn't projecting very well. I'm the lead of the musculoskeletal theme now, so overseeing a lot of support for all the different areas within musculoskeletal. But I also lead the vasculitis workstream, and that's what I'm going to focus on a little bit today. So in terms of all of us together within musculoskeletal, there's a lot of parallels with the type of research that we're doing between all the different musculoskeletal diseases. So, and we're all at different stages of that journey. Vasculitis is very new to the BRC, so we're at a lot earlier stage in the journey than with some of the other conditions. But one of the big aims that we're trying to do is to identify people who are at risk of developing either inflammatory diseases or musculoskeletal disease, to either prevent the disease itself. And for some diseases, I think we're looking at the potential for trying to prevent the disease occurring, and that's where there's early signs such as antibodies we can pick up in the blood. There's a real chance of trying to be able to do that. Or for other conditions that we don't have the antibodies, maybe thinking, the focus is on preventing complications of these conditions. Another component that cuts across a lot of the inflammatory diseases is trying to reduce the side effects of drugs. And one drug that most of us use is steroids or prednisolone. And, many of you may have had these. We use them to different amounts in different diseases, but really want to try and focus on trying to reduce the side effects from steroids. And a lot of that is finding alternative drugs to steroids and reducing that added burden of the other diseases that happen once you get some sort of musculoskeletal disease. And I think one thing we're all very much aiming towards is thinking about how we can target different treatments to different individuals depending on their the type of features of the disease they have, or something like their genetic background or the particular complications. How can we make treatment more tailored? In order to do that, we need to have several drugs that we can use to treat the disease, and some of the different conditions we don't have many drugs that have been shown to work. So part of that is finding new drugs and new treatments. So that's more or less what I was going to say about the theme as a whole. So for my own vasculitis work stream, this is co-led by myself and Dr. Mark Iles, who's a statistical geneticist. And as I'll come to show you later, I think to really make progress in a lot of these conditions, we need to be looking at big data sets, and it's absolutely vital we have really good statisticians and statistical support to be able to make the most of the data. And I will show you some of the different data sets that we're starting to get access to us now to use for research. And for this BRC, we tried at the outset when we put in the application to set a grand challenge of something that we really wanted to aim towards. And I think whilst the work stream broadly covers vasculitis, I think steroids are the absolute hallmark of the one of the main drugs that we use to treat vasculitis, and we have to use big doses, so people do get side effects. And so if we can find ways of treating the condition with significantly reducing the steroids, I think the outcomes will be better for everything. And we've done lots of work talking to people with lived experience of a whole range of inflammatory diseases, and versus arthritis have done the same. And it's what we get back all the time of find something else other than steroids. So I don't want to say that they're not good drugs, because you have to have an alternative, but actually if we can find those alternatives, I think we'll make huge inroads into quality of life for people. I know not everybody's necessarily going to have a vasculitis in the room. So it's what that essentially means. It's an inflammatory condition that occurs in blood vessels. So when the blood vessels get inflamed and cells from the immune system move into the walls of blood vessels, that's what we would call a vasculitis. And the way we try to, we sort of talk about the different types of vasculitis is it's down to the main size of the blood vessels involved. So here you can see if it affects the main blood vessels coming down from the heart and supplying the arms and legs. We talk about that as a large vessel vasculitis because it's the larger blood vessels of the body. If it's the blood vessels going into the kidney or into one of the other organs of the body, we'd refer to that as a medium vessel vasculitis. And then if it's the tiny blood vessels within the kidney, within the inside of the kidney, or the tiny blood vessels within the skin, we talk about this as a small vessel vasculitis. And there's probably 60, 80 different types of vasculitis. We've got a huge number of different types of vasculitis, but we tend to divide them into these three main categories. For some people, they just have vasculitis, one of these types of vasculitis. Other people get it secondary to an infection or get it as part of another condition such as rheumatoid arthritis. So there's a whole range of different conditions that cause it. I'm going to start with giant cell arteritis. It's one of the types of vasculitis that I've done the most research on, and so I know there's quite a lot of people in the room that have got this condition. I can go on afterwards and cover different types of vasculitis. So this is a condition which affects the large and the medium blood vessels. So thinking of those blood vessels coming from the heart, going to the arms, the leg, the big. So it's, as I say, it's a type of vasculitis that affects the large and predominantly the large and the medium blood vessels. And we recognize different forms. So if you'd have asked me what giant cell arteritis was 10, 15 years ago, I'd have described a condition where people would get headaches, they would have inflammation of blood vessels that affected their head and their neck, and potentially blood vessels that went to the brain or to the eye. And some of those people may have had polymyalgia as well. And that's what you might hear us say now as cranial GCA. So it's all the blood vessels supplying the head and neck. And that's the very typical type of of giant cell arteritis. What we also recognise, and we probably started to recognise about 10, 15 years ago, is actually what we hadn't been detecting in many of the people with giant cell arteritis, that you can also get involvement of the aorta, the blood vessels supplying the arms and the legs. And we refer to that as a large vessel vasculitis. And so there are these different patterns of which blood, even within one type of vasculitis, which blood vessels are affecting different people. What we can also, and if people who have just the large vessel vasculitis, when this wasn't recognised and wasn't treated, what happened is that could weaken the wall of the blood vessels within the aorta, and people started to develop aortic aneurysm. So there's some big series from the Mayo Clinic, which is where this wasn't picked up and wasn't recognised, and people did start to get the aneurysms. So with the picture, this is when we used to do more temporal artery biopsy. This is what we see when we look under the microscope. You get immune cells starting to move into the vessel wall, and this very inner part there, just beyond the white, just in this section, is where the blood would normally flow, and you get so many immune cells moving into the vessel wall, it blocks the blood flow. So what we can see with people, and some of you may have had this, the blood vessels become very tender, they can become very thickened if they're within the scalp, but also you can block the blood supply. And if that happens in the skin, you can get changes within the skin. But if it happens with the eye, you can then get visual symptoms, or you can get loss of vision from this. And this is the main biological process. It's all these immune cells moving into the vessel, and then the wall of the vessel reacting to those blood cells. What's really changed things is now recognizing that some people have what we call a large vessel vasculitis as well. And we've got lots of fantastic new imaging techniques now. So some of you will have had an ultrasound scan that Dr. Sarah Mackey's been very pioneering in setting up in Leeds. But we also have other imaging scans where we can start to see if the larger blood vessels are inflamed. A subgroup of people have a related condition, polymyalgia rheumatica, which is what you asked me about. So not everybody with giant cell arteritis has polymyalgia rheumatica. This huge debate, is this a spectrum of one disease? Are they two diseases that happen to occur together? And there's different views. I think ultimately genetics, and when we look at the biology, that's really going to tell us. I've got my own very personal views on this, but, you know, I think there is this question of whether they're the same or different diseases. And other people get these really profound inflammatory symptoms. They may lose weight, they may have sweats, and are incredibly unwell at the time. So all these different things we call the same condition. And this is just an example that I was saying, you know, some people with giant cell arteritis will just have the blood vessels of the head and neck involved. Some people might get that to a certain extent, but it's other blood vessels that tend to be involved. And then this is a condition we see in children and young adults called Takayasu arteritis. And this is almost identical to what we see with large vessel vasculitis. So there's some subtle differences between the two, but these two look very close together. But within medicine, you know, there's various definitions of diseases that have evolved over the years based just on clinical findings. And I think, as I come to show you with some of the genetics, I think as time goes on, what we call different diseases may change over the years, and it may be that the treatment we use for this disease should be the same as this one, and not that, even though we call them both giant cell arteritis. So in terms of giant cell arteritis, when you sort of think about the treatment challenges, and this is, I suppose, thinking of it of both sides. For giant cell arteritis, the standard treatment is to start steroids. And if you think about it, everybody's over 50. A lot of people are in the 70s and 80s, may have other conditions. We need to use very high doses. So this does result inevitably in side effects. It's really important that we use steroids because at the moment we don't have anything else that prevents the problems like the blindness and the other symptoms. So I don't say we shouldn't be using them, but we don't have many alternatives. The problem is we reduce the dose down, a number of people can relapse, and some people need very big doses of steroids and can need them over many years. I've got some people who've needed treatment for 10, 15 years. So it's not necessarily a condition that burns out. And we see a lot of side effects. And the other problem until relatively recently is we had very few trials. So the trials that have been done in the standard drugs that we use in rheumatology, none of those seem to work. We haven't had good trials in leflunomide, and I think that's a pity. We've never been able to get those funded. But relatively recently, we had, a few years ago, we had a new drug licensed, one called tocilizumab, and we're now able to prescribe this, and I'll come on to that in a little bit more detail at the end. And we've got other drugs in trials, and I think the big problem with giant cell arteritis and clinical trials is a lot of the pharmaceutical companies, there was an anxiety about giving a drug and reducing the amount of steroid when there was a risk of blindness. And I think it was because of the concern over the risk of blindness that then made people very reluctant to then fund clinical trials. Once you get the first trial design that it's is safe and is acceptable, and you've got a drug going through, it just paves the way. And so I think it's a really exciting time now because there's lots of drugs going into clinical trials, but it's always getting that first clinical trial. And for this particular one, there was one clinical trial, and they designed it in such a way they went straight to licensing, and they got that approved by the FDA. So I think there are treatment challenges. So to just say a little bit, I'm going to come back to the tocilizumab again, but I'm just going to say a little bit about steroids. So, as I say, steroids have side effects, but they've also transformed medicine as well. And, it was in 1950 that it was in the sort of 1940s that the first clinical trials of when steroids were discovered, and there was the first clinical trial, and the first disease that steroids were used to treat was rheumatoid arthritis. So there's been a long-standing experience of using steroids in rheumatology. First approved in 1955, and it's still on the FDA or WHO list of essential medicines. And one thing that's going to become relevant later, it's a cheap drug. It costs pennies. A course of steroids might be costing hundreds of pounds a year. We're not into the sorts of costs that you would have with other drugs, but they are very good drugs. And large numbers of the people in the UK, so this was a study that Dr. Mackey led a number of years ago, 1% of the population in the UK are taking these treatments, 3% of people over the age of 70. So these are widely used drugs across a whole range of diseases. And we see quite significant side effects from these. So, there are a number of side effects. I don't want to dwell on those today. what I absolutely don't want anyone to do after this talk is to say, well, I'm going to stop my steroids. Because if you've been on steroids for a long period of time, your adrenal glands may not be producing sufficient steroids, and we have to very slowly wean people off steroids. And so it always has to be that discussion with your doctor. We can aspire to find other drugs, but there's a really important safety issue of don't just suddenly stop steroids, because actually we need your adrenal glands to start making steroids again. So for me, this is a really, really important slide about tocilizumab. So this is a slide from the clinical trial. And this looks at people after a year of treatment to say how many people have stayed in complete remission, no evidence of a flare of their condition over a year of treatment. This group of people, so this is for giant cell arteritis, this group of people had 6-month course of steroids. This group of people had a 1-year course of steroids, and this group of people had a 6-month course of steroids and tocilizumab, which is one of the new drugs we have available. And you can, so 56% of people who had tocilizumab, in addition to that initial course of steroids, actually had no flares over that year. So huge improvement in quality of life. And on the basis of this trial, and I know there's some people in the room who've had tocilizumab. Initially, when it was reviewed by NICE, it was turned down, and there was a lot of lobbying to get access to it. We're now in a situation where we can prescribe it for a maximum of 1 year once in a patient's disease journey. So we can't give a prolonged course of treatment. We can prescribe it once, and not to everybody. There's certain subgroups of people who can have the tocilizumab. I'm actually meeting the NHS England commissioning lead again on Thursday to sort of discuss this. As you can imagine, I've been lobbying hard on behalf of everyone. And the reason for this is because what we're comparing it to is steroids, and steroids don't, are worth, it's a few £100s for a year. And when this drug was first released, it was maybe, I don't know, £12,000–15,000 a year. So the amount of data available at that time on the costs of the steroid side effects was, there wasn't that much data there. So when you do a health economic argument, the only way we could get this approved, and because the trials only lasted a year, they said you can have a year for subgroups of patients. So it's come off patent now. The costs are going to be coming down in a few months' time. And so we're sort of putting that pressure on again in these discussions. So I think we're now in a very different position because, a few years ago, we had drugs that we were trying, we used in other branches of rheumatology that may work in individual patients, but now we're starting to get much better drugs. And if you think of trying to always be improving treatments, still at the beginning, the way our guidelines are working is that we just start people on steroids and no other drugs. But then, sorry, I was going in the wrong direction. We're now at the point that we can start to give steroid sparing drugs for high-risk patients but again, for only a year. But I think what we really want to be getting towards is that tocilizumab available to all. Where it's indicated, there might be reasons why it wouldn't be right, but, where we feel it needs to be used. And that's something, we're all lobbying very hard for. But it will be fantastic, and it might be tocilizumab isn't the right drug for this, to be able to find alternatives with a whole range of treatments. And then once we get several, the ideal is you can start to say, well, actually, this is your pattern of disease, whatever it is, we think you would benefit from one drug or another. So going back about 6 or 7 years ago, we realised the only way we were going to make any advances in giant cell arteritis was to get a big group of people nationally to work together. So I approached all the leaders who were working in this area in the UK, and we all came together. So there's an absolute UK-wide approach to this, and we had a whole number of different studies. This was a relatively small amount of money just to set up the consortium in giant cell arteritis. And I think what we've done out of, I think we've got about £700,000. We really have done a huge number of studies. And I just want to say a huge thank you to Diane, who's been one of our the sort of public partners and patient partners on, and she's been helping us sort of think about giant cell arteritis for many years. And you know, we've involved local charity groups. I've maybe not come towards the local group quite so much, because quite a long time ago we founded this big national group. And one thing that, when Dr. Mackey was my PhD student many years ago, and I know quite a lot of you see Dr. Mackey as a consultant now, we formed the UK GCA consortium, and that was part of Sarah's PhD. We're now in a position, we've recruited over 3,500 people nationally with giant cell arteritis. I think we have about 70 centres recruiting nationally at the moment, and we've kept it really simple. And it's mainly a genetic study, and we're really starting to get some really exciting insights with that. So there may be another session we sort of talk through a little bit about that study. But this is something I wanted to bring out. So one of the big work streams of this grant many years ago was trying to understand about steroid side effects, because unless we could say how many people got it, what the risk was, and then work out the costs, we were never going to get steroids from being beyond costing £200 a year. If you can cost the impact of the side effects, suddenly you can balance up with NICE and the way they do their assessments, that actually steroids start to cost more than one of the new drugs. So it's, you know, it's in some ways it's what you know, we all know, but actually unless you have the data, unless you've got the publications, NICE can't consider it. And so this is one of my colleagues who was at Leeds now. She's a very senior statistician in one of the pharmaceutical companies, and fantastic epidemiologist. And we went to a huge public resource of primary care records. And you might see this from your GPs, and you're happy for your data to be used for research. And we access this through the clinical practice research data link. So as you can see, this was done quite a long time ago in 2017. And by doing this, is just using routinely collected data from GPs, we found nearly 90,000 people who'd had steroids for an immune-mediated inflammatory disease. I'm not going to go through how we did all the analysis, but we were just sequentially went through a whole number of the complications and worked out what the excess risk was of steroids. So we had to take into account people's age and other medical conditions, but what's the excess risk? And I think it doesn't matter. This is for infections, all-cause infections, and it was every type of infection. I could plot high blood pressure, heart disease, diabetes. We see this strong dose response that it doesn't matter. This is if you're not using steroids, you're on up to 5 milligrams, 5 to 15, 15 to 25, and more than 25. So you have this dose response that so this is why I'd say there probably isn't a safe dose. The question is whether this level of risk is worth it for an individual to have that better quality of life if there isn't an alternative drug. So I don't want to completely demonize them, but actually by doing this systematically for most of the side effects, most of the common side effects of steroids, we've published it all. And so the hope is this is now in the public domain for NICE to use. And I'm working with a health economics team in Leeds to try and redo those NICE models. And that's what we're aiming to do. So we can then, and I think if the cost of tocilizumab comes down, we can show steroids are more expensive than the drug itself. We might get that spot where we can actually prescribe tocilizumab for everyone. So I think if we're going to, think about how we find new treatments, then there's a whole number of ways that we can do that. We can sometimes borrow drugs from different conditions. So, and that's what we've done historically, thinking, well, this drug works for rheumatoid arthritis, or it works for a different condition. Shall we try it in this disease? So that's one approach. But that approach hasn't been that successful in giant cell arteritis. A lot of the trials weren't particularly good. Another place we can start is starting to think about what someone's genetic makeup are, how the immune system works, and the environment. And for a lot of the immune and the inflammatory diseases, it's this mixture between your genetic makeup, the environment, and it for some diseases, it could be risk factors like smoking. For some, it could be pollution. In others, it could be simply infections, that you've been exposed to infections. And some of it is how your immune system responds. And the fact for some of these conditions, we have, they happen as people are getting older. It might be the aging of the immune system. So your body, you know, if you've got a certain genetic makeup, you're prone to get these conditions and then as your immune system starts to age, you just lose that little bit of control of the immune system. So it's likely to be a combination of all of those. And so when we set up the UK GCA consortium, we needed to do something really simple, something, you know, relatively low cost. DNA is very stable, and, you know, on the whole, is stable throughout someone's course. So it meant we could collect data from people, and it didn't matter what time, you know, where you were in your disease journey. And so when there's a whole number of ways we can do this, and there's just a very simple code with genetics. You've got 4 different bases or nucleotides, and we refer to them as A, T, G, and C. And all of the proteins and all of the makeup in your body is just made up of this really simple genetic code. And they're constantly, as you get all these different changes. Some of them are really common in the population. So you will get these small changes, and they might make some people more resistant to certain infections. So for the immune system, actually, it's the infections that have molded our genetic makeup. So we can look at the DNA, which is what we've done a lot of. We can then look to see, well, in individual cells, they take different fragments of the DNA, and that then gets made into something called RNA. So that's what we call, it gets transcribed, so it gets made into another form. And then that leads on to proteins. So one thing we've done from temporal artery biopsies of people who've been part of our research studies is you can go to those biopsies from the NHS pathology labs and actually take out RNA from those and understand what processes been turned over. So I've got a PhD student working on that at the moment. And then from blood samples, we can look at proteins. We can look at all the proteins that are in blood. So we can really start to build a map of the biology. So we've done a lot of genetic studies. This is one study which, you know, we look at them in individual diseases, but actually what you can do is put a whole number of different diseases. So this was all the genetic studies of all the different types of vasculitis. If we put them all together, are there some common pathways that we see in every different type of vasculitis? Don't worry about all the different types. But what we get with a genetic study is a plot like this, and this is what we call a Manhattan plot. And up this axis here is just the p-value. And we just set an arbitrary cutoff if it's of where it's statistically significant. And with these studies, what we will do is we'll type 500,000 or 1 million different genetic variants, and then we can statistically expand that out. So we've got 5 million different variants. So there'll be some we can predict what they're going to be, even if we haven't typed them directly. So you might have an individual 5 million different genetic variants, and we just line them all up, chromosome by chromosome. So each of these little dots is a different genetic variant. And we create these plots, and then you start to see these little peaks of particular genes that are overrepresented in people with a particular condition compared to controls. It's a numbers game in genetics. So if you put all the different types of vasculitis together, you see them. And what we're starting to see here is this is a particular immune mediator. And there's a drug that's used in inflammatory bowel disease that targets that pathway. This is something related to how T cells function. And we've got a whole number of drugs that we can use to target that particular immune cell. This, again, is something that's very relevant to T cells. So you can start to see from the genetics, if you've got large numbers, actually, we need to be looking at that cell or that cell. It's never going to give you the whole answer, but it's just those clues as to what's the most important. So we've got this. I don't want to say too much because I don't want to blind you with charts, but actually, I think we're getting huge numbers now. And we've got 3 genetic studies that have been submitted at the moment that are making us rethink about what we need to be looking at in terms of giant cell arteritis. The other thing that you can also do is look at the proteins in the blood, and the proteins are under a lot of tight regulation. But also you get a bit of leakage from the blood vessels, you get a bit of leakage of proteins from the liver, and what we saw with COVID, because obviously with the pandemic, there's this huge amount of trying to understand what the biology was, why people were getting the side effects. And what we found is actually looking at the proteins gave us more information quickly than a lot of the genetics and the other things. And this was just making the point, if we know which cells are involved, we've got long lists of drugs that are in clinical trials for other types of vasculitis or we're using for other diseases. So it gives us some real pointers. So I think where next, what we're going to be doing next? Well, I think we do have now where some of our lab studies are really giving us clues for new targets and new treatments that we can start thinking about. I think we really need to improve how we predict who's going to relapse. So one study that we're trying very hard to do in Leeds, it was difficult during the pandemic, is for anyone who's having tocilizumab, trying to get blood samples from people when they're on tocilizumab and established on it, and then take a sample about 6 to 8 weeks after people stop tocilizumab, and then take one if people flare later. And what we're trying to understand is can we predict who's going to flare just with when people are on tocilizumab, or can we predict for when we have that change in proteins over that first few weeks of stopping, if someone's going to flare? And so there's lots of studies like that we're trying to do locally. A lot of that will be looking at the proteins in the blood. And as I say, doing a lot of work with the health economists, because there's no point trying to find new drugs for these conditions if we can never get them approved through NICE. So I think we have to do both together. And as I say, medicine's never been more exciting. I mean, the wealth of data that we have is just enormous at the moment. And it's really important, I think, that, there has to be the ethics and the governance. But I've always been very collaborative around the UK. No data will get shared, no personal data. But unless we work together and collaboratively, we're never going to make progress. We'll never have enough people with this condition. And that's why I set up the genetics study across the whole of the UK. And I think it's really important to train the next generation that, and we desperately need the statisticians, the data scientists, and we need the next people to come and be part of that team. Probably won't go through all of this. There's a whole number of other studies. There's big national investment in genetics. And so you've heard of things like UK Biobank, which we use. I lead the vasculitis studies for the NHR Bioresource, and I think we're the highest recruiter in Leeds for that. So thank you for that. And we also were working very closely with our Future Health, trying to think of how we can do much more risk prediction. And the last thing I wanted to mention is all the BRCs come together and work together on a number of projects. And there's this group called the Translational Research Collaborative. There's one across all the musculoskeletal diseases, and this is co-funded by the NHR and Versus Arthritis. And I think a lot of the lobbying I've been making about steroids was really listened to, and we're just setting up a whole new work stream now on steroid toxicity, and that goes across adult and childhood diseases. It goes across the rheumatic diseases, and we put out an advert across the BRCs. We've got about 60, 70 people around the UK involved, and we've got ophthalmologists, rheumatologists, nephrologists, cardiologists, endocrinologists. And I think it's been fantastic. So we will be writing further grants, because I think what we learned from one condition, it's important we can go to the other. And I think the most important thing is the strategy for this has to go to the Department of Health, and you have to get ratification by the Department of Health. So by lobbying, and as I say, a lot of the lobbying started around the tocilizumab argument, we can then start to have those funding calls that will allow us to really try and identify new drugs. And I'm sorry if it's been long. I think it's been a little bit, I wasn't intending it to be this long, but it's been a bit stop-start with the projector and things. So I don't know whether anyone's got any further questions, and I'm happy to chat informally at the end.
Do we know how many people are diagnosed with GCA and LVV?